Microbiology, along with my colleague, Professor Vandana Luthra, who is from Department of Physics, Gargi College, extend a very warm welcome to all the participants, that is faculty and students, to the today's talk on antimicrobial nanoparticle resistance phenomena in pathogens by Dr. Cindy Gunawan. Now, this talk has been organized under the aegis of National Academy of Sciences India Science Society program at Gargi College, University of Delhi. For this, I would like to thank Professor Ajay Ghatak, who is President of National Association of Sciences of India, and Professor Anurag Sharma, who is Vice President of NASI Delhi chapter, and for providing us this opportunity to conduct these diverse activities and doing these wonderful, so interesting webinars. And then I actually we all have got immense support from our principal, so I'm obliged to our principal, Professor Promila Kumar, who has been giving us constant support and encouraging us to undertake such activities. Our ma'am could not join due to her busy schedule, but her best wishes are with us. Now, coming to the introduction to speaker of the day, Dr. Cindy Gunawan, I would request Professor Vandana to introduce her to the audience.
Thank you so much, Professor Shashi. It is indeed my privilege and honor to welcome and introduce Dr. Cindy Gunawan to all. Dr. Cindy Gunawan is a senior lecturer and leader of Biofunctional Nanomaterials Group at the Australian Institute for Microbiology and Infection, University of Technology, Sydney, UTS. Dr. Gunawan is a nanobiologist with expertise in the fundamental and translational research of nanoparticle cell interactions. Her research group studies the characteristics and molecular basis of bacterial, toxicological, and adaptation responses to nanoparticles, focusing on a range of pathogens in both their free-living, planktonic, and surface-attached biofilm forms. Through her nanobio expertise, Dr. Gunawan also contributes to the development of smart nanoparticle-based antimicrobials, as well as studies on the environmental and human health impact of nanoparticles. These pioneering research, including the discovery of the phenomenon of bacterial resistance to the nanoparticle, have been published and highlighted in prestigious journals such as Small, ACS Nano, and Nature Nanotechnology. Dr. Gunawan received her Bachelor of Engineering with the University Medal in 2002 and her PhD in Biotechnology in 2006 from the University of New South Wales, that is UNSW, Sydney. She had her postdoctoral training in the School of Chemical Engineering in UNSW and was then awarded the UTS Chancellor's Research Postdoctoral Fellowship in 2015. Dr. Gunawan is the lead chief investigator for Australian Research Council Discovery Projects as well as Australian Academy of Science Projects. Now, I would hand over the mic to Dr. Cindy for her much awaited talk on bacteria adaptation to nanoparticle toxicity and how to overcome it. There is a little bit change in our talk, I think, in the title, but the overall gist of it is going to be the same. So over to you, Dr. Cindy.
Thank you, professors, for your introductions. So yes, good evening, everyone. I'm Dr. Cindy Gunawan from the Australian Institute for Microbiology and Infection in UTS. So today I'm going to share our work on antimicrobial nanoparticles to see how these antimicrobial nanoparticles targets bacteria, and in turn how bacteria can develop resistance or adaptation phenomena to the nanoparticle, and then what can we do as a scientist to try to overcome these resistance phenomena. So we're focusing our study on silver nanoparticles. You might ask, why nanosilver? Silver nanoparticles is an ultrafine, less than 100 nanometers nanoparticulate. It has different forms, so you can have metallic silver and also silver 1 oxide. So research inquiries have determined that these silver nanoparticles have broad-spectrum antimicrobial activity. So it has shown efficacy on bacteria, yeast, and also viruses. And as such, silver nanoparticles is now considered as one of the most important alternative antimicrobial to antibiotics. So because of this broad-spectrum, well-known broad-spectrum activity, silver nanoparticles have been incorporated in medical devices, such as here in wound dressings and here in the catheters. But because of its lucrative values, many companies have now also incorporated silver nanoparticles into consumer products here. So for example, in personal care products such as toothbrushes, soaps, hair dryers, even in household and kitchen appliances, here in washing machines, fridges, microwaves, and even in baby products. So this widespread and indiscriminate use of nanosilver has given us a concern, because we know that extensive exposure of microorganisms to antibiotics have led to the development of bacterial resistance to antibiotics. So we are worried that the same thing could happen with the silver nanoparticles. So before we got into how the bacteria, as whether or not the bacteria can develop resistance to silver nanoparticles, let's have a look on how these nanoparticles exert antibacterial activity. So this is a cell envelope of bacteria. Here is the gram-positive bacterial envelope, and this is the gram-negative bacterial envelope. So silver nanoparticles, when it's exposed to aqueous medium, it will partially release soluble silver ions. And during this dissolution process, a reactive oxygen species will form. And these reactive oxygen species can then target the cell envelope.
Cindy,
yes?
Dr. Cindy, sorry to interrupt, but your slides are not being changed, I think.
Which one do you see at the moment?
First slide only.
Oh, okay. But it's changed here. Oh, hang on. So now it's the first slide, right? Like the green ones.
Yes, yes. Now is the first slide.
Okay. How about now? Is it the second slide?
Not yet. At least not yet seen to me.
Oh, okay.
Anyone else can write in the chat if they are able to see the second slide.
No, now the second one has come, Vandana. Can you see the second now?
For me, I think it is still stuck. I don't know.
Can you see the second slide?
We can proceed.
Yeah, we can proceed. I think everybody is able to see.
Yeah. Now it is visible. Yeah, now it is visible.
How about now? Can you see in the title it says how nanosilver exerts antibacterial activity?
Yeah, perfect. Now we are on the second slide now.
Okay.
Yeah, you may proceed. Thank you so much.
Okay. So we're going to have a look on how nanosilver exerts antibacterial activity. So this is our bacterial cell envelope. Here is the cell envelope for the gram-positive bacteria, and this is for gram-negative bacteria. So when the nanosilver, when it's exposed to aqueous environment, it will partially release soluble silver ions, so leach a soluble silver species. So during this dissolution process, there could be a formation of reactive oxygen species. And we have shown that these reactive oxygen species can damage the cell envelope. So here is our Fourier transform infrared spectroscopy or FTIR studies on the effect of nanosilver on peptidoglycan. So peptidoglycan is a polymeric matrix composed of polymers of amino sugars, and between these polymer of amino sugars, it is interlinked by a peptide chain. So this is the amino sugars here. It will alternate between N-acetyl glucosamine and N-acetylmuramic acid to make a polymeric amino sugar chain, and then this amino sugar chain will be interlinked by this peptide chain here. Now, peptidoglycan forms the outermost layer of gram-positive bacteria, and a thinner layer of peptidoglycan is also present in gram-negative bacteria here between the outer membrane and the inner membrane. So our infrared spectroscopy have shown that exposure of peptidoglycan to silver nanoparticles actually disrupts the amide groups that are present in the peptide chain, as well as in the amino sugars. So this actually disrupts the whole structure of peptidoglycan. It breaks the interlinking between the amino sugars layer, and we also found that the nanosilver also breaks this beta-1,4 glycosidic bond between the amino sugars. So,
Dr. Cindy, sorry to bother. Your slide is stuck at the second one.
the second one, which one?
It's why nanosilver.
Oh, okay. How about this one? Could you see the hang on just a minute. Okay.
Hello, Cindy.
Yes.
Just stop sharing the screen just for once.
Okay, I'll just stop it first. Okay, yep. Okay, stop sharing now.
Now share again.
Okay,
click on share.
Okay, how to go there? Okay, I'll try again.
Share. Now select the whole PC. You might be getting multiple options. Instead of that, you use share screen. Whole screen will getting some option like that.
I have this entire screen window and
You
Yes.
click on entire screen window.
entire screen. Okay. Share. And
now switch to the PPT, please.
Yeah. Yeah, I see. How about now?
It's loading. Just a minute.
Okay. Can you see it now?
No, we don't see it yet. Is the PPT running?
Yeah, it is running.
Okay. Actually, we are not able to see. Can you minimize the screen? We are.
So what did you see at the moment?
We are seeing your Chrome browser page.
Okay. Okay, hang on. Okay, I think the screen, entire screen, share. Okay, now I go to here.
Yes, now the PPT is visible.
Yep, how about now?
Yes, visible right now.
Okay. Yep.
Yes,
Yeah.
it's moving.
Yeah, it's moving.
Okay. How about I just, because it's just 2 slides, I think I can just start from the beginning if you want.
Yeah, it will be better for our students. Sure.
That's okay. I think I know why, because I think I have two WebEx opened.
Yes, right.
Okay, now it's okay, right?
Yeah, it's moving now. Perfectly all right. Yeah.
Okay, so just start from the beginning. So this is so again in these presentation, I will share with you our research focus on the antimicrobial nanoparticles. How specifically on how these nanoparticles target bacteria, and in turn how the bacteria develop adaptation phenomena to the nanoparticles, and then what we can do as a scientist to actually overcome the resistance phenomena. So we are focusing our study on the silver nanoparticles. Silver nanoparticles is an ultrafine, less than 100 nanometers particulate of metal oxide and metallic silver. Now, studies have shown, many studies have shown that nanosilver has potent and broad-spectrum antimicrobial activity. So it is effective against bacteria, yeast, and even viruses. And because of this, nanosilver has been widely considered as an important alternative antimicrobial to antibiotics. So because of these, as a result of this potency, silver nanoparticles has been incorporated in medical devices, such as here in wound dressings, as well as in catheters. Because of its lucrative values, companies have also been incorporating nanosilver into consumer products. Here in personal care products, such as in soaps, toothbrushes, toothpaste, in kitchen and household appliances, in washing machines here, and in fridges, as well as in baby products. Now, these widespread and indiscriminate nanosilver applications has been worrying, because we worry about that just like the case in antibiotics, this extensive use of nanosilver can eventually lead to a resistance development by bacteria, just like in the case of antibiotics. So in this presentation, I'm going to show you that yes, the bacteria has this capability, natural ability to develop resistance to silver nanoparticles. But before we go into that, let's have a look on how the silver nanoparticles exert antibacterial activity. So again, this is our bacteria. These are the cell envelope of bacteria. This is the cell envelope of a gram-positive bacteria, and this is the cell envelope of a gram-negative bacteria. So how does nanosilver targets the cell envelope? Silver nanoparticles, when exposed in aqueous medium, will leach soluble silver. And during this dissolution process, there could be a generation of reactive oxygen radicals, or ROS. And we have shown that this ROS can cause oxidative damages on the cell envelope. Here with our ATR Fourier transform infrared spectroscopy, or ATR-FTIR, we have shown that silver nanoparticles can disrupt the peptidoglycan layer in bacteria. So peptidoglycan layer is basically a polymeric chain of amino sugars. So it is an inter-alternating polymer chain of N-acetylglucosamine and N-acetylmuramic acid being linked by these beta-glycosidic linkages, and that this polymeric chain of the amino sugars is interlinked by the peptide chain. And our infrared spectroscopy have shown that nanosilver can actually disrupt the amide groups that are present in the peptide chain here, as well as in the amino sugars here. Nanosilver can also disrupt these beta-1,4 glycosidic linkages that link the amino sugars. So here with our infrared spectroscopy, we've shown that the nanosilver can target the peptidoglycan layer here in the gram-positive bacteria, as well as here the thin layer of peptidoglycan in the gram-negative bacteria. We further found that nanosilver is not just attacking the peptidoglycan layer in the cell envelope. Nanosilver also damage the phosphatidylethanol amine. It is the phospholipid groups that are present in the inner membrane of gram-positive bacteria here, and in the outer membrane and inner membrane of gram-negative bacteria here. With our infrared spectroscopy, we could see that the nanosilver damages the phosphate head groups of the phospholipids as well as the fatty acid tail of the phospholipids. So these damages that are caused by nanosilver on the cell envelope, in particular those on the phospholipids that compose the inner membrane, will disrupt the respiratory chain components that are embedded in the inner membrane of bacteria. And as a result, there will be a disruptions of the electron transport chain from the respiratory chain components, such that we will have leakage of electrons into the cytoplasm. So these electrons then reacts with molecular oxygen to produce the superoxide radicals, the reactive superoxide radicals. And these superoxide radicals can in turn attack the iron sulfur clusters in proteins, releasing the Fenton active Fe2+, and this Fe2+ can then react with cellular hydrogen peroxide to produce the highly damaging hydroxyl radicals. So this formation of cellular reactive oxygen species, or ROS, can then cause oxidative damage on proteins, lipids, and DNA. So cellular studies that show these cellular ROS generations in bacteria. So on the top panel here, this is a fluorescent microscopic images of the cell-only control, so no silver, and we dye the cell control, the bacterial control, with a fluorescent dye that can detect cellular presence of reactive oxygen species, which will give us green fluorescence. And as you can see here with the control, we could only detect very low levels of reactive oxygen species, which is most likely coming from the byproducts, which is most likely a byproducts from the aerobic respiration. And as a result, or in agreement, we only detect low amount of dead cells in the control, shown here by the red cells. So what happened if this bacterial bottle is exposed to silver nanoparticles? As you can see here, the exposure of the bacteria to the nanoparticles lead to the rapid generation of lethal levels of cellular oxidative stress, as you can see here with the green cells. So here, even only after 5 minutes of exposure, we could detect high levels of cellular ROS in the cells. And consequently, we also observe high levels of dead cells. So up to this stage, we have seen how the silver nanoparticle targets the bacteria and how that these antibacterial mechanisms is complex and multi-targeting. So there has been a common perception that it would be difficult for bacteria to develop resistance to such complex and multi-targeting toxicity. But in 2013, our group discovered for the first time the development of bacterial resistance to silver nanoparticles. So this is our study in 2013. So I'm just going to have one slide on this one, on our 2013 study. We found the phenomena of bacterial resistance to nanosilver in an environmental group of bacteria called Bacillus. So this Bacillus, following prolonged exposure to nanosilver, was able to develop a stable resistance trait to silver nanoparticles, such that after the prolonged exposures, the bacterium was able to proliferate at an otherwise toxic nanosilver concentration. And since our discovery in 2013, there are other scientists who have discovered, who have also confirmed this development of bacterial resistance in other bacteria. In 2015 here in E. coli, and in 2018 here in Pseudomonas aeruginosa. Both bacteria are gram-negative bacteria and they are clinically important. Then we ask ourselves a question. So these E. coli and Pseudomonas aeruginosa, they have this presence or prevalence of seal genes in their genomes. Seal genes is a set of 9 genes that code for proteins that of which function is to efflux, is to get rid of silver ions from the cells. So we ask ourselves this question: how about if a bacteria who does not have seal genes in their genome, can they also develop resistance to silver nanoparticles? So that's the motivation of why we actually embarking the new study of silver nanoparticle adaptation phenomena in this bacteria, the Staphylococcus aureus. So Staph aureus, as you know, is a gram-positive bacterium. There is only a very low prevalence of seal genes in the Staph aureus. Staph aureus has been considered as one of the priority pathogens because of its ability to develop resistance to multiple antibiotics. And Staph aureus is also one of the leading causes of hospital-acquired infections. So let me take you through the process of how these bacteria, without any presence of seal genes, can develop resistance to nanosilver. So we subjected the bacteria to prolonged exposures of nanosilver, here for 50 days. And in this study, we're not just studying nanosilver, we also do a comparison, a parallel study with ionic silver, as well as with the antibiotic ciprofloxacin. So the study with the antibiotic ciprofloxacin here, it's not just to compare the potentially different adaptation responses of this bacterium to silver in comparison to antibiotics, but also as a validation of the experiments, because we already know that Staph aureus can develop stable resistance to ciprofloxacin. So this is what we do. We expose the bacteria Staph aureus over 50 days of continuous prolonged exposures to silver nanoparticles through sequential passaging or subculturing every 24 hours. And you could see it here as you proceed with the prolonged exposure, you could see that the bacterium has developed this ability to grow and grow at progressively increasing nanosilver concentrations. The same thing also happened. The same thing were also observed for the ionic silver here. Such in the course of 50 days exposure, the bacterium were able to progressively proliferate, even when it is subjected to progressively increasing concentration of ionic silver. And of course, with the ciprofloxacin, it is expected. So we have this ability. We've seen this ability of the bacterium to proliferate at really high concentration of the antibiotics. So here we could see that it should be that this bacterium were able to proliferate at increasing concentration, under exposures to increasing concentration of the antibacterial agents. So this bacterium must have developed some kind of adaptation responses. Now, to validate for a resistance development, what we did is that we took these cultures at the end of the prolonged exposures. We then grew these cultures on an agar plate that contained the respective antibacterial agent. And then we picked the resistant colonies, or colonies that have been growing on those antimicrobial agar plates. We then culture these colonies for several days in an antimicrobial free liquid medium. So no silver, no antibiotics for several days. The reason for this is just we would like to isolate for a cell population that has developed stable adaptation phenomena to these antibacterial agents, and excluding any subpopulation of the bacteria that only have a transient adaptation responses. After the antimicrobial free culturing, we then retested the minimum inhibition concentration of each of the antibacterial agent on the passage bacteria here. So this is a result for the Nanosilver. So the dotted line here is so you can see, so this is the profile of bacterial growth profile in the presence when exposed to increasing concentration of nanosilver. The dotted profile here is the growth profile for wild-type bacteria. It means the bacteria that has not been pre-exposed to silver nanoparticles. And as you can see that the minimum inhibition concentration of nanosilver on the wild-type strain is about 3 micrograms per liter. 3 micrograms per mil. So minimum inhibition concentration is the smallest concentration of an antibacterial agent to inhibit the growth. And here for silver nanoparticles, we could see that the MIC, or the minimum inhibition concentration, is at 3 micrograms per mil for the wild type. So what happened with this MIC? With the bacteria that has been previously pre-exposed to nanosilver. So we could see that the MIC of the pre-exposed bacterial population has increased to 15 to 20 micrograms per mil. So it means that there is a 5 to 7-fold increase in the MIC of nanosilver on the Staph aureus, which confirms for the development of a stable resistance trait. How about for ionic silver? For ionic silver, we also observed a 3-fold increase in the MIC. And of course, for the ciprofloxacin, we observed a significantly high, more than 64-fold MIC increase for the passage bacteria. So from this experiment, we can conclude that Staph aureus can develop stable resistance trait to silver nanoparticles, and that this resistance and extent of resistance is quite modest compared to the antibiotics. So then we bring our study further. We would like to understand, okay, the Staph aureus, it does not have any seal genes. The set of genes that conferring resistance to ionic silver. So how does this Staph aureus develop resistance to the silver nanoparticles? So to get on the bottom of this, we did a whole genome sequencing to compare the whole genomes of the cultured bacteria to the wild-type strain, of the passage bacteria to the wild-type strain, to find for potential single nucleotide mutations. Now, we found mutations on a gene called the POR gene. So POR, basically a gene that encodes for proteins that regulates the purine nucleotide synthesis. Okay, let's have a look. So under normal conditions, this POR protein will sit on a specific DNA binding site on the purine operon, such that it will prevent the RNA polymerase from transcribing this purine operon. So upon prolonged exposure to nanosilver, we detected a mutation in POR protein, gene, and therefore on a POR protein, on a specific site of POR protein that binds onto this specific binding site, such that we hypothesize that these POR mutation causing will lower the binding affinity of the POR protein to this specific DNA site, and therefore will allow the RNA polymerase to transcribe the whole purine operon. And indeed, we have confirmed this with our RNA sequencing studies that confirm the upregulations or the higher expressions of all the genes in the purine operon. So you might now ask yourself, okay, what does this have to do with the nanosilver resistance? Why does this Staph aureus is mutating its POR gene to fight off the nanosilver toxicity? What's the connection? Now, nanosilver has been known to target DNA and proteins in Staph aureus. This purine nucleotide is important for the DNA replication, DNA repairs, as well as for the synthesis of amino acids in Staph aureus. So we hypothesize that this Staph aureus is mutating its POR gene such that we will have higher expressions of this whole purine operon, such that we will have more purine nucleotide to serve as the building blocks for DNA repairs and also for the amino acid synthesis to fight off the nanosilver toxicity, because nanosilver is targeting the DNA and proteins in Staph aureus. So next, I'm going to share you two more gene mutations that we observed in the nanosilver resistance, or in this case, ionic silver resistant Staph aureus. So apart from the POR gene mutations, we also observed mutations in the TCYA and CIMR genes. This gene encodes proteins that are involved in cysteine metabolism. So how does these mutations actually relate to resistance development? So to understand this, we need to go back. We need to recall on how the nanosilver is targeting the bacteria. So if you recall, the silver can attack the iron-sulfur clusters in proteins, releasing the iron 2+. The iron 2+ reacts with the cellular hydrogen peroxide to produce the reactive oxygen species. Therefore, a silver-mediated oxidative stress. So how does the cysteine comes into picture? Now, cysteine is an amino acid. It is actually the building block for this sulfur in the iron-sulfur clusters. Cysteine is also one of the building blocks of thioredoxin. Thioredoxin is an antioxidant molecule in gram-positive bacterium, such like Staph aureus, that can scavenge the reactive oxygen species. So now you're asking yourself, okay, cysteine is so beneficial for the Staph aureus to try to fight off the silver toxicity. So why does the bacteria introducing mutations on genes that are involved on cysteine metabolisms? Now, recent research inquiries have shown that the cysteine can reduce the iron 3+ in the cells to produce iron 2+. And again, the iron 2+, it can react with hydrogen peroxide to produce ROS. So this is called a cysteine-mediated oxidative stress. So these mutations in the TCYA and CIMR gene is thought to try to mediate or to try to avoid or minimize this cysteine-mediated oxidative stress. TCYA gene encodes TCYA protein, And the function of the protein is to import extracellular presence of L-cysteine, not cysteine, L-cysteine, which is a precursor for L-cysteine. So these mutations on the TCYA gene and therefore on a TCYA protein is thought to try to limit or to control the import of the extracellular L-cysteine into the cells, such that to guard the cellular cysteine levels at safe levels to avoid the cysteine-mediated oxidative stress. And CIM-R gene here encodes for CIM-R protein, which regulates this process that is mediated by the TCYA. So here you can see that the bacterium can develop a mutation that can introduce physiological changes on its cellular pathways to fight off, to counteract the nanosilver toxicity. Now, we have the opportunity, apart from doing the DNA studies, DNA level studies to look for gene mutations, we have also had the opportunity to do studies at the RNA levels and then at the metabolomic or metabolite levels. So don't worry about the complicated scheme. I just want to highlight that these major cellular pathways are found to be modified in the Staph aureus as a result of the prolonged exposure to nanosilver. So we thought that these cellular pathways are being affected when the bacteria is trying to defend the nanosilver toxicity. So we found changes in the iron sulfur synthesis pathways. And of course, this is understandable, this is logical, because iron sulfur clusters is one of the targets of nanosilver. We also found upregulations in the antioxidant enzymes in the bacterium to try to scavenge off reactive oxygen species. We also found observed an upregulations of an efflux mechanisms by to get rid of the ionic silver from the cells. So up to this stage, we have seen how the nanosilver targets the bacteria. It's complex, have multi-targeting toxicity, and then in turn how the bacteria can develop resistance to this complex multi-targeting silver nanoparticle toxicity by changing its genomes, by introducing gene mutations. So the next important question would be, okay, we observe this in the laboratory settings. Will this happen in the real life settings, in the real life application of nanosilver? So to investigate this, to get insights into this, we investigated over 400 products that contain nanosilver. And we pay attention, we're focusing on the pathways as whether these products can facilitate the prolonged exposure of microorganisms to bioavailable silver, which we have known so far it is key for the bacterial resistance development. So in this study, for the products, we have a look on how the silver is being released from the products, whether there is a possible systemic absorption of the bioavailable or toxic silver into the bloodstream, and ultimately the silver distribution and accumulation in organs and tissues. And with this, we also have a look or taking into account of the possible chemical transformation of silver. So from this study, just quickly, we found that there is a genuine potential for silver, for accumulation of toxic silver in organs and tissues that are inhabited by microorganisms. Because of the use of nanosilver containing product in real-life applications, such that here in urinary tract, on wounds, gastrointestinal tract, mouth cavity, as well as in the respiratory tract. So from up to this stage, we have seen, okay, bacteria can develop resistance to silver nanoparticles, and indeed there is a genuine potential for this to happen in a real-life application of nanosilver. And therefore, we arrive to my last part of the presentation. What can we do as a scientist to try to minimize this tendency or overcome the resistance phenomena? So we have this chance to create or to develop a smart antimicrobial nanoparticles. To play tricks on bacteria, we tuned the physical-chemical characteristics of nanosilver, in this case the oxidation state, with a simple photo technique to try to manipulate the antimicrobial activity on bacteria to disrupt these prolonged exposures of bacteria to nanosilver, which we have known to be key in the bacterial resistance development, to switch on and off the antimicrobial activity of nanosilver. So how to do this? To do this, we doped the silver 1 oxide, nano-sized silver 1 oxide on titanium dioxide. So the silver 1 oxide here is about 2 nanometers-ish. This titanium dioxide support is about 30 nanometers. And this is the electron microscopy images of these particles. You could see the big blob of titanium dioxide here and a small nano-sized deposits of AG1 on the surface of titanium dioxide. So you might be wondering why titanium dioxide? Because we know that titanium dioxide is a semiconductor. It means that it is responsive to UV light. So upon irradiation of these particle composite to UVA, because of the band gap of the titanium dioxide, there will be an excitation of electrons from the valence band to the conduction band here, valence band to the conduction band of titanium dioxide. And in turn, these electrons will reduce the Ag1 deposits on the surface of titanium dioxide to metallic silver. And as you can see here, with increasing UVA irradiation time, you could see this presence, more prominent presence of surface plasmon resonance, or SPR, at between 450 to 550 nanometers, which is a characteristic of nanometallic silver. So this occurrence of surface plasmon resonance, this hump here, confirms that the silver 1 deposits has been reduced to metallic silver. Now, because we have metallic silver here, right? Now the silver 1 has been converted to metallic silver. And we know that metallic silver, because of its surface plasmon resonance, will now be responsive to visible light, of which wavelength higher than 450 nanometers. So upon irradiation of these particles with visible light, what happens is that because you already have the metallic silver deposits on the in the particles, so there will be a reverse electron flows from the silver deposits to the titanium dioxide, and in turn the metallic silver is oxidized back to silver 1. And to confirm this, with the visible light illumination, we could see this disappearance of surface plasmon resonance. It means that the metallic silver here has been oxidized back to silver 1. So you might be asking questions now. Okay, so we have used this photo switching technique to manipulate the oxidation state of silver. But how can this actually translate to the manipulation of the antimicrobial activity? So we use a model bacteria to test this. We use E. coli as our model bacteria, and we expose the E. coli to these particles. So first, let me show you the switching off of the antimicrobial activity of nanosilver. So this is the growth profile of the E. coli bacteria with respect as a function of time. So the blue profile here is the growth profile when the model bacterium is exposed to the nanoparticles without any elimination. So as prepared. If you remember, you will have a silver one deposit here. As you can see, the growth of the bacteria is completely inhibited. But when you expose the bacteria to nanoparticles that has been exposed to increasing time of UVA elimination, you can see more and more growth, meaning less and less growth inhibition, which means that the antimicrobial activity has been switched off. So the switching off of the antimicrobial activity actually matches with the leaching of soluble silver. So here we could see at 0-hour UVA elimination, it means that as prepared nanoparticles without any UVA elimination, you could see about 38% of leaching here. And when you increase the UVA pre-irradiation, you could see less and less release of soluble silver into the system. And as you know, soluble silver that are released from nanoparticles is also toxic. So we have seen how the antimicrobial activity has been switched off. But now, can we actually switch on and off the antimicrobial activity of nanosilver? So this is the one that we have before, the as-prepared nanoparticles without any UV elimination, and this is the growth profile when the nanoparticles has been exposed to UV elimination, which means that the silver one deposit has been reduced to metallic silver, and therefore the switching off of the antimicrobial activity. And this is the corresponding extent of leaching of the nanoparticles. 0-hour UV, you have high extent of AG leaching, and then with 40 hours of UV elimination, we have low extent of silver leaching. Okay, how can we switch? Okay, these nanoparticles have been switched off. So how about if we take these nanoparticles and then expose that to a round of visible light illumination? Yes? Yes? Can I continue?
Yeah. Please continue.
Okay. So how about we take these nanoparticles and then expose that to a round of visible light illumination. If you remember from the scheme, with the UVA illumination, the silver one has been reduced to metallic silver. And because it has been reduced to metallic silver, which these nanoparticles is responsive to visible light because of its surface plasmon resonance. And therefore, when we expose these nanoparticles to visible light, the metallic silver deposit will be oxidized back to silver 1. And when we test with the bacteria, we could see that the growth profile is inhibited again. It means that the antimicrobial activity is switched on. And as a result, this is the corresponding extent of leaching of soluble silver from the nanoparticles. You could see with another round of visible light illumination, we could see again the leaching of soluble silver jumped again into the high levels. Okay, lastly, we have switch off and switch on, and we would like to see whether we can go a full cycle of switching off the antimicrobial activity again. So we took these nanoparticles, we exposed that to another round of UVA illumination, which means that the silver one deposit is switched back to is reduced back to the silver, a metallic silver, and accordingly, we could see this switching off of the antimicrobial activity, which this growth profile. And in line with this, we could see a drop in the leaching of soluble silver. So up to this stage, I didn't show it here, but we did expose our model bacteria to this switch on and off nanosilver. And it did delay the bacterial resistance development, but the resistance phenomena ultimately occurred. And it's just delaying. It didn't actually solve the problem. But this actually showed that how we can use, it actually showcased how we can modulate the nanoparticle antimicrobial activity by tuning characteristics for an intended biological functions. In this case, trying to delay or minimize the tendency for resistance. As an extra slide here, I would like to show you. This is the switch on nanosilver. As you can see, with the switch on nanosilver, it caused a complete growth inhibition. We could see high level of cellular rust generation here and high level of dead cells. And this is the switch off nanosilver. You can see that the silver wand deposits is reduced to metallic silver. We could see only, or we can detect only low levels of cellular rust and therefore low levels of dead cells. Therefore, the switching off. So it's in agreement with the switching off of the antimicrobial activity. So this ends my presentation. So to conclude from here, we have seen how the bacteria can remarkably develop resistance to this complex multi-targeting toxicity of nanoparticles. Our next mission is to bring this study even further to the next level to study the adaptation phenomena in biofilm form of growth, because biofilms, as we know, is the major form of microbial growth. So, as a take-home message, there is this genuine potential of bacterial resistance development in real-life application of nanosilver. So we need to be careful, to be very judicial with the use of antimicrobial nanoparticles, including nanosilver, just to use it when they're needed. This is to ensure the successful long-term use of this important alternative antimicrobial agent. So that ends my presentation. I would like to acknowledge all my collaborators from UTS and also from the in Australia and also from overseas. And I would like to also thank the Gunawan lab team, our postdoc and students, and also the Australian Research Council and the Australian Academy of Science for funding our research. Thank you. So you can contact me here.
Thank you so much. Thank you so much, Dr. Cindy, for such an informative and enlightening talk on different aspects of silver nanoparticles and its interaction with the bacteria. And that was really good. And I would like to tell to the students that other than the basic contribution to the literature, these kind of studies have industrial value also, and quite significant industrial value because of their possible use and their continuous use in the medical and many other fields. And so many products, like you showed that 400 products use silver nanoparticles. So I would like to ask before the forum is open for the question, like, what are the stringent, are there not stringent conditions that how much and what kind of these nanomaterials can be used in the daily use products?
Yeah, so there has been regulation surrounding the use of nanoparticles. So it's not just antimicrobial nanoparticles. and each country has their own regulations. For example, here in Australia, we have it's called a NICNAST, and I think there is one for European Unions and also in the US. There has been regulations, but at this stage, of course, because nanomaterials, it's not really new, but it's still a developing field, in particular with the use of the antimicrobial nanoparticles. So these regulations, it's not at this stage, there are still loopholes with these regulations. So for example, in Australia, silver nanoparticles, for example, is being considered the same as the box silver, the micron size, not the nano size silver. So the nano size silver, because of their similar composition, is still classified as silver, not silver nanoparticles. So this has subjected loose reporting requirements, assessment requirements in terms of toxicity and so on with the silver nanoparticles, because as you know, although they have similar composition, silver on oxide or metallic silver, the micron size silver or the box silver has much less reactivity compared to the nano size silver. So that's one of the one example of the loose regulations that we have at the moment. So our study, and also with other scientists, is trying to inform strategies, scientific-based strategies, to try to develop a more effectively regulated use, not just use, but also the production, the management, and disposal of antimicrobial nanoparticles, in particular with the realization that bacteria can also develop resistance to these nanoparticles, which is quite, if it's just been found in 2013, it's not even a decade. And although there are these emerging studies by other scientists confirming the development of resistance and bacteria to these nanoparticles, I think what happened is that we are still gathering more evidence, a more concrete scientific evidence, such that we can inform the right strategy surrounding the use of this potent antimicrobial agent. Yeah, so to answer your questions, so far, as you know, there has been a lot of use of, for example, on silver nanoparticles in consumer products because of that loopholes in the regulations. Nanosilver,
right, absolutely.
it's being considered as silver, and it's not.
Absolutely so.
And that's why we need the work of scientists to try to figure it out, more evidence on the reactivity or microbiological activity of these nanoparticles, in particular with the bacterial defense side, which is emerging at the moment.
Yeah, we also had done a small project with undergraduate students long time back, and that was in association with the microbiology department. Actually, I am from physics department, and we had prepared these nanoparticles using mulberry tea leaves, and in that, when we varied the ratio of the that extract to the silver, and then we could tailor make the sizes of that, and depending upon the size, we had a difference in the antimicrobial activity. And that we could show that we can tailor make the sizes, and that has got a significant effect on the antimicrobial activity. So in your case, what was the size of the nanoparticles? Like, did you use a standard protocol for that, or did you vary the size also?
We actually, from the beginning, from the start of our nanosilver work, we only use one type of nanoparticles, that the silver doped titanium dioxide that we had, that was presented, that was shown in the slides. So we have stick with that kind of nanoparticle composite since the beginning, because our study, when I moved to the Australian Institute for Microbiology and Infection, our study, it's getting deeper and deeper into the molecular biology of bacteria and how to understand how the bacteria responses or develop defense to these nanoparticles. So we try to keep our nanoparticle the same because, yes, you're right. If you have different size, you have different shape, you have different surface functional groups, that it will change the extent of the antibacterial activity. So far we have stick with that kind of nanoparticles. We have about 2 nanometers nanosilver deposits on the top of titanium dioxide, and that's a good question also because titanium dioxide is photoactive. It is responsive to UV, and as you know, you have 5% UV in our ambient light. So we always perform or carry out our experiment in dark conditions to try.
In that also,
Yes.
in that also, I think there are very many advancements because people have tried to use visible light by doping, like it becomes active to the visible light in case of
Yes.
TiO2, and
Yes.
then that also can be explored that because instead of UV light, then the visible light can also be used. And
Yes.
another question would be a comment that the silver mediated oxidative stress you have talked about in the silver nanoparticles. So
Yes.
this amount will change in the case of magnetic ZNO and other nanoparticles. So that would also be a very interesting study to just
Yes.
compare the nature of the nanoparticles with that of, say,
Yes.
silver.
Yep. So it's our study in 2018. We try to get into the bottom line of the fundamental antimicrobial mechanisms of the nanosilver, because as we know, regardless of the size, shape, and surface functional groups, there are always 2 main components of the silver nanoparticle active agents. There is the silver particulates and the leach soluble silver. So in 2018, we found that both the silver particulates that remain after leaching and also the leach soluble silver both can contribute to the overall antibacterial activity of silver. Because I think there has been a belief that it is only the leach-soluble silver that are microbiologically active, while the silver, the solid part, is inert or benign. But we shown these are we actually shown the otherwise, that actually both are microbiologically active. And this is when the effect of size, functional group, and shape, comes into, have an even more prominent role because of the microbiological activity of the solid particulate itself. And then also the size, shape, and surface functional group will also govern or will also affect the degree of soluble silver leaching from the nanoparticles. So yeah, it is quite a complex antimicrobial mechanism. Yes. But it is very potent at the same time. If you compare with other metallic nanoparticles, that you notice that you actually need a much less per mass basis to exert the same extent of antimicrobial activity.
Yeah, before I will share another result, there is a question by Avya, and she wants to ask that, can we use silver nanoparticles for cancer therapy?
Yeah, of course, for the cancer therapy, there has been studies on that, but of course it does not dwell on the antimicrobial activity of the silver nanoparticles. I think it dwells more to the use of silver nanoparticles as a delivery vehicle for some agents to try to target the cancer cells. But yes, there has been studies on that.
Yeah, I would like to add to this that can I add?
Yeah.
Yes, Avya, in this case, for the cancer therapy, normally hyperthermia technique is used, and in that, the magnetic nanoparticles are also in use for that case.
Yep.
And here I wanted to share one result with you, Dr. Cindy, that we had doped magnetic nanoparticles with different dopants, and we saw that after a particular doping, it changes its morphology from spherical to rod shape. So
Okay.
that would also be something that can be seen in terms of its efficacy towards these studies.
Yep.
Okay, so that was just a comment. And now we have Dr. Irshad Ahmed, who is also asking that what is the exact mechanism of anti-biofilm activity of silver nanoparticles in case of S. aureus?
We for S. aureus, we haven't actually got into the biofilm form. With the study that we have so far with Staph aureus, it's still in a planktonic state. But we have actually did some detailed study with Pseudomonas aeruginosa, it's in particular with the strain PA01, which is known to form extensive biofilms. So this is what we found. The silver nanoparticles actually can't inhibit the growth of the Pseudomonas aeruginosa biofilm. So it actually preventing, or it has this growth inhibition activity. So it when we put because we know that this bacteria can form biofilms, so we actually expose the bacteria at the same, with the nanosilver, and we found this quite a reasonable minimum inhibition concentration of the nanosilver on the biofilm growth. We also found that the nanosilver can eradicate a mature or a pre-grown biofilm. So this is called minimum biofilm eradication concentration, or MBEC. So we know that nanosilver at this stage can indeed inhibit the formation of biofilm.
Just a moment. Please unmute yourself. Somebody's mic is on. Please, all of you are requested to kindly keep it off. Yeah, please continue.
I think she didn't realize. Yeah. Okay. So
yeah, please continue.
Yeah. So with the nanosilver, we know that it can inhibit the growth of the biofilm. So before it's forming, it inhibits the growth, and it can also eradicate a pre-grown biofilms. So with the exact activity, on how this nanosilver can inhibit or eradicate a pre-grown biofilms, we're still investigating it, but we know that, for example, the nanosilver can actually penetrate these biofilm layers. Yep. Okay. Hello.
Yes, Dr. Cindy, somebody's mic is on, so really apologies for that. Yeah.
That's okay.
Please continue. Please continue.
Yeah, so we know that the nanosilver can inhibit. I think it's still on.
Yeah, it's still on. Can we request Mr. Anush to kindly make the mic off of the person who is speaking?
So
please continue.
So the nanosilver can actually inhibit the growth and also eradicate a pre-grown biofilm of Pseudomonas aeruginosa. How exactly that nanosilver can do this? With the inhibition, I think it is, as we can consider it as a normal activity of the targeting of nanosilver. It attacks the cell envelope, it produces reactive oxygen species, and so on. But the curious thing is that how can the nanosilver eradicate the pre-grown biofilms? Because to do that, the nanosilver or the leach-soluble silver need to penetrate those thick protective EPS layer of the biofilms. But we have shown that nanosilver can indeed eradicate that biofilm growth. We've shown that up to a certain level, the silver nanoparticles can actually penetrate the thick EPS layer and reaching the cells underneath. But of course, we still need more investigations to see, as you know, after this nanosilver penetrating this thick EPS layer of biofilms, whether it can still exert its normal activity because of the presence of the EPS and other organic moieties that won't be present with the planktonic cells.
Thank you so much. And I think this field is such an interdisciplinary field that you have an option of manipulating at the nano level, as well as you can see the microbiological intervention into that, and that can be basically explored for core shell and other kinds of nanoparticles also with the help of surfactants or without surfactants. So there is a question by Dr. Professor Shashi here, that how our students can join your institute?
How? Sorry, how?
How our students can join your institute? What should they do if they want to join your institute?
Or the students?
Yeah, our students, yeah.
Yes, so we UTS, it needs to be UTS through international program. So UTS every year opens this international scholarship for postgraduate studies. So both master and PhD studies. So that's through UTS. But me also having a research grant. So we are currently looking for 2 PhD students that will work on the multi-species biofilms to understand how the silver nanopart how the multi-species biofilms, in this case a mixed culture of Pseudomonas aeruginosa and Staph aureus, can develop adaptation to silver nanoparticles. So the PhD the 2 PhD positions are on that study. To basically understand the extent of adaptation phenomena, what type of adaptation phenomena, because we can have resistance, tolerance, or persistence, and how does it work at the molecular level, to understand the bottom line of it. So with that research grant, because it is my own group research grant, so any interest can be emailed to me. So yeah, basically there are 2 ways. From the international scholarship program that was being organized by UTS central level. So to do that, if you're interested to do a PhD with me, you need to email me, and basically email me, and then we can work out whether we need to apply to the UTS international program or applying for my research group scholarship.
Oh, that will be wonderful if our students can join your institute.
Yes,
so I would also like to be, I'll be keen to ask that they should have a basic degree in microbiology or in any sciences, like that would be the precondition for getting into PhD. Do they need to have a graduate degree in physical sciences or life sciences, or it can be any?
It could be basically we already have postdoc PhDs from different backgrounds, but yes, preferably to have some degree, the degree is related to microbiology somehow. So for example, I have already have PhD student, or she has finished. So she is from the biomedical engineering. So it's not really microbiology, but has some biology in it. And of course, Riti is from, it's having a microbiology degree, and some others, for example, are from chemical engineering with a little bit of biology background. But yeah, so we like someone. So basically, from any background with maybe a little biology in it, but I think we're looking for someone who has great initiatives and like independent and have great initiatives, and which is and who is a team member. Yes, just like a job application.
Thank you so much for your insights into these all these aspects with such a great detail. I cannot see any other question right now, and I assume that all the questions you have answered so beautifully. And with that, I can only request everyone to please switch on your cameras for a while so that we can have a photograph. May I request Dr. Cindy to you as
Yes,
well?
yes,
So
yes.
that will be a great memory with us. And with that, after that, I will hand over to Professor Shashi for the vote of thanks. Thank you so much.
It's already nighttime here.
But we will have a great memory of having you with us. So please.
You're still muted, Professor.
Professor Shashi, you need to unmute yourself.
Yeah, okay. Actually, I was saying that if you
So,
can click the picture because my net is not working. Suddenly, कुछ फिर से गड़बड़ हो गई है.
Can you send me
okay.
Can you send me the photo too?
Yeah. That will
Sure,
be
sure,
our
we will do that.
pleasure.
We will do that.
But I am getting only 6 people in the, like, I'm not having the full layout. So first let me click one of these and
Yeah.
then I'll do the next one.
There are 37 participants.
Yeah.
How to do? I don't know how to do. Because I never
Yeah,
used
that's okay.
I never used
Students can also take, I think we can also request the
Yeah.
students. Yeah, they can
Yeah.
also take side by side
Third
on
years,
chat.
आप लोग हो यहाँ पे, please take 1 or 2 pictures. Whosoever is on the laptop, please take it.
And do visit India also. We invite you for Indian visit also. It will be our pleasure.
Yes, I will. Thank you.
Yeah, sure.
Yeah.
Because our students will be highly elated to meet you. That is, and including us also. So that will be something which they will always cherish for their lives. So yeah, I have taken one. Yeah,
Yeah.
and I hope some students have also taken. Yeah.
So we'll send you some.
Okay, thank you.
Yeah.
Maybe I'll I don't know how to. Okay.
You just have to print screen and you will get the picture.
Okay. See, with technology.
Yeah. So are we done, Vandana? For the picture?
Done. Yeah.
Okay, so
I hand it over to you now. Yeah, for the final vote of thanks. Thank you so much, Dr. Cindy. Thank you very much.
You're welcome. Thank you.
Yeah, thank you for the thank you, Professor Vandana. So it is my pleasure and privilege to propose this vote of thanks for today's talk. First of all, I would like to thank our principal, Professor Pramila Kumar, for her immense support and inspiration for organizing this webinar. My heartfelt gratitude to our esteemed speaker of the day, Dr. Cindy Gunawan, for accepting our invitation and giving such an insightful talk on nanoparticles. I found it so simple. I mean, you made it so simple for us to understand. And it was really very systematic. I mean, I'm really impressed with the kind of presentation
Thank you.
we had, it was really nice. So
Thank
we are
you.
sure that this session has enlightened our students, faculty members, and other participants about this concept. And then the thing is that this opportunity was provided to us by the National Academy of Sciences, India. I and Professor Vandana are thankful to the Academy for allowing us to conduct these diverse activities for the benefit of society under this unique Science Society program. I'm also grateful to our mentors, Professor Ajoy Ghotak, who is president of NASI India, and Professor Anurag Sharma, who is vice president NASI and chairperson of the NASI Delhi chapter. So thank you both the sirs for the kind of support they've been giving us. Then special thanks to officials at NASI Prayagraj for all the administrative support. Then a special mention of Mr. Anuj for technical support. He's the person who keeps helping us and whenever we are stuck somewhere in the conducting the webinars and all. So technical support is provided by him and lab staff for administrative help. And finally, a great thank to all the participants who have attended this webinar for sparing their valuable time for attending this talk. So thank you everyone for being here with us today at this time. Thank you everybody.
Thank
So,
you.
Thanks,
thank you,
Dr.
Dr.
Cindy.
Cindy.
Especially
Yeah.
it is night, so we can't thank you enough in by our words.
Yeah.
Thank you
I
so much.
really appreciate you and thank you, and hoping to see you sometimes in India.
Yes.
We
Yeah.
will.
Sure.
Yeah.
Thank you.
Okay,
Thank you,
bye-bye.
everyone. Bye-bye. Bye-bye.
Bye. Bye,
Thank you,
everyone.
everyone. Thank you for
Bye-bye,
organizing this.
everyone. Yeah. Bye, everyone.
Thank you.
Anuj, we can close the meeting now.